So hi, Nana Divya. Last class I took botany summary boxes. அதில் வந்து செல் டிவிஷன் செல் சைக்கிள் அப்புறம் செல் யூனிட் ஆஃப் லைஃப் மைக்ரோப்ஸ் லெசன் மட்டும் கவர் பண்ணல. அது வந்து இந்த ஜுவாலஜியோட சம்மரி பாக்ஸில் வந்துட்டு கவர் பண்ணிட்டேன். ஸோ லெட்ஸ் டோன்ட் டிலே, அப்புறம் கிளாஸ் ஸ்டார்ட் பண்ணிடலாம். ஸோ எஸ்என்ஜி டெஸ்ட் சீரீஸ் பற்றி சொல்லணுன்னா இட் ஹாஸ் லைக் கான்செப்ஷுவல் பேஸ்ட் டெஸ்ட் சீரீஸ், அப்புறம் கிராண்ட் டெஸ்ட், லைக் ஒரு லெசனோட லைக் சாப்டர் வைஸ் கொஷின் பேங்க்ஸ் எல்லாமே இருக்குது. ஸோ வந்து மேக் யூஸ் ஆஃப் இட். வெறும் 500 மட்டும்தான் அது. இந்த மாதிரி ஆஃபர்லாம் நாங்கள் படிக்கிறப்போ கிடையாது. ஸோ மேக் யூஸ் ஆஃப் இட். எல்லோரும் ரெஜிஸ்டர் பண்ணுங்கள். ஆஃப்டர் சீயிங் எவ்ரி வீடியோஸ் போய் அந்த டெஸ்ட் சீரீஸ்லாம் அட்டன் பண்ணிங்கன்னா லைக் வெரி யூஸ்ஃபுல். ரொம்ப நல்லா இருக்குது கொஷின்ஸ் எல்லாமே. லைக் லாஸ்ட் இயர் டாப்பர்ஸ் எல்லாம் எடுத்த கொஷின்ஸ் தான் இருக்குது மோஸ்ட்லி. So let's start with the lesson biological classification. Biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological characteristics. So then Linnaeus later classified all the living organisms into two kingdoms, Plantae and Animalia. So Whittaker proposed an elaborate 5-kingdom classification: Monera, Protista, Fungi, Plantae and Animalia. So first வந்து plants and animals வந்து அரிஸ்டோல் வந்து ஸோ பேசிஸ் ஆஃப் மார்ஃபாலஜிக்கல் கேரக்டரிஸ்டிக்ஸில் வந்துட்டு கிளாஸிஃபை பண்ணியிருப்பார். ஸோ எக்ஸ்டர்னலாக அது எப்படி இருக்குது அதை பேஸ் பண்ணி கிளாஸிஃபை த ஆர்கானிசம் ஆஃப் பிளான்ஸ் அண்ட் அனிமல்ஸ். அதுக்கப்புறம் வந்து லினரஸ் என்ன பண்ணாருன்னா 2 கிங்டமாக பிரிச்சிட்டார், பிளான்ட்டே அண்ட் அனிமலியா ஸோ எல்லா அனிமல்ஸையும் தூக்கி அனிமல்ஸில் பாக்டீரியா அதெல்லாம் தூக்கி வந்து பிளான்ட்டில் போட்டுட்டாங்க. ஸோ தென் விட்டக்கர் தான் என்ன பண்ணார், ஈ ப்ரொப்போஸ் எலாபரேட் 5 கிங்டம் கிளாஸிஃபிகேஷன். ஸோ 5 கிங்டம் கிளாஸிஃபிகேஷனில் வி ஹவ் மொனீரா, புரோட்டிஸ்டா, ஃபங்கி, பிளான்ட்டே அண்ட் அனிமாலியா. The main criteria of 5 kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationship. So in the 5 kingdom classification it doesn't only depend on the morphological characters. So more than that, it has the cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationship. In the 5 kingdom classification, bacteria are included in kingdom Monera. So bacteria are cosmopolitan. So Monera kingdom வந்துட்டு we have bacteria. Bacteria are cosmopolitan in distribution. Cosmopolitan they occur everywhere. That's cosmopolitan. These organisms show the most extensive metabolic diversity. So it has very diverse metabolic. Bacteria may be autotrophic or heterotrophic in the mode of nutrition. So it has both autotrophic and heterotrophic mode of nutrition. So bacteria வந்து 2 types இருக்கு, Archaebacteria and Eubacteria. Archaebacteria and Eubacteria. So Archaebacteria வந்துட்டு they develop on harsh conditions. ரொம்ப ட்ராபிக்கல் ஏரியாஸ், தெர்மோஃபைல்ஸ், அசிடோஃபைல்ஸ் அந்த மாதிரி ஏரியாவில் தான் வந்து ஆர்கேபாக்டீரியா இருக்கும். ஸோ யூபாக்டீரியா ஒரு எக்ஸாம்பிள் வந்து சைனோபாக்டீரியா. ஸோ சைனோபாக்டீரியா இஸ் வெரி யூஸ்ஃபுல் இன் நைட்ரோஜன் ஃபிக்ஸிங். தென் கம்ஸ் கிங்டம் Protista. Protista includes all single cell eukaryotes such as chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. ஸோ ப்ரோட்டிஸ்டாவில் வந்து சிங்கிள் செல் யூகேரியாட்ஸ்லாம் இருக்குது. ஸோ அதில் என்னென்ன குரூப்ஸ்லாம் வருதுன்னா கிரைசோஃபைட்ஸ், டைனோஃப்ளாஜிலேட்ஸ், யூக்ளினாய்ட்ஸ், ஸ்லைம் மோல்ஸ், அதுக்கப்புறம் ப்ரோட்டோசோவன்ஸ் எல்லாமே இருக்குது. ஸோ கிரைசோஃபைட்ஸோட எக்ஸாம்பிள் பார்த்தோன்னா டைஅட்டம்ஸ் அண்ட் கோல்டன் ஆல்கே. இந்த டைஅட்டம்ஸ் அண்ட் கோல்டன் ஆல்கே எப்படி இருக்கும்னா they float passively in water. இதை நம்ம என்ன சொல்லுவோம்னா பிளாங்க்டன்னு சொல்லுவோம். Then comes dinoflagellates. So dinoflagellates வந்து they appear in different color like red, blue, green அந்த மாதிரி depend on the pigment on them. So red color dinoflagellate வந்துட்டு we call it as goniolax. அதுக்குள்ள வந்துட்டு it has 2 flagella. Then euglenoids. Euglenoids ஓட எக்ஸாம்பிள் வந்துட்டு euglena. So euglena has pellicle-like structure instead of cell wall. It makes its body more flexible. So euglenoids are very flexible. Then comes the slime molds, then protozoans. Protozoans, there are 4 types: amoeboid protozoan, flagellate, ciliated, and sporozoans. Then protists have defined nucleus and other membrane-bound organelles. So other membrane-bound organelles, it comes the lysosome, endoplasmic reticulum, and the membrane-bound organelles. Then they reproduce both asexually and sexually. Members of kingdom fungi show great diversity in structure and habitat. So most fungi are saprophytic in nature. So fungi are mostly saprophytic in nature. Their mode of nutrition, they show asexual and sexual reproduction. So in fungi, asexual reproduction is by zoospores, conidia, and sporangiospores, and sexual reproduction is by oospores, ascospores, and basidospores. Phycomites, Ascomycetes, Basidomycetes, and Deuteromycetes are the 4 classes under this kingdom. Then the plantae includes all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, pteridophytes, gymnosperm, angiosperm are included in this group. So இதெல்லாமே வந்துட்டு we saw elaboratedly in the plant kingdom about algae, bryophytes, pteridophytes, gymnosperm, and angiosperms பத்திலாம். The life cycle of plants exhibit alteration of generation, gametophytic and sporophytic generation. The heterotrophic eukaryotic multicellular organism lacking a cell wall or included in kingdom Animalia. So Animalia, they include the multicellular organism that lacks cell wall. The mode of nutrition of this organism is holozoic mode of nutrition. They reproduce mostly by the sexual mode. Some acellular organisms like virus, viroids, as well lichens. Lichens and all, it is symbiotic association between the algae and the fungi. So these are all not included in the 5 kingdom system of classification. So this is the 5 kingdom classification. ஸோ எல்லா கிங்டமோட செல் டைப், செல் வால், நியூக்ளியர் மெம்பரேன் ப்ரெசண்ட்டா ஆப்சென்ட்டா, அதோட பாடி ஆர்கனைசேஷன், மோட் ஆஃப் நியூட்ரிஷன், இதெல்லாமே வந்துட்டு we need to know. ஸோ முனீராவில் வந்து இட்ஸ் ப்ரோக்யூராட்டிக், இதில் சிங்கிள் செல் யூக்கேராட்டிக், அண்ட் தென் இது எல்லாமே யூக்கேராட்டிக் ஆர்கனைசம். ஸோ யூ நீட் டு நோ த கிளாஸிஃபிகேஷன் ஆஃப் செல் வால், ஸோ அதர் திங்ஸ் ஆர் ஆல். ஸோ அவ்வளோதான் பயாலஜிக்கல் கிளாஸிஃபிகேஷன். ஓகே, தென் we go to animal kingdom. The basic fundamental features such as level of organization, symmetry, cell organization, coelom, segmentation, notochord have enabled us to broadly classify the animal kingdom. So animal kingdom, எப்படி வந்து like we classify animals பார்த்தோன்னா, its level of organization, symmetry, cell organization, coelom, segmentation, notochord. So coelomனா it is body cavity, notochordனா it is a rod-like structure present on the dorsal side. Besides the fundamental features, there are many other distinctive characteristics which are specific for each phylum or class. So, எல்லா phylum or classல வந்துட்டு their specific characters hold for that particular class alone. So, எப்படி classify பண்ணியிருக்காங்கன்னா levels of organization, symmetry, body cavity, and then phylum. So, this is very important. எதுக்கு cellular level, எதுக்கு tissue organ or organ system, எது coelomate, எது acoelomate. So, இதெல்லாமே வந்துட்டு we need to know. First comes the porifera. Porifera includes multicellular animals which exhibit cellular level of organization and have characteristic flagellated conocyst. Sonocytes are known as collar cells. It covers the spongiocele. Spongiocele is the body cavity of porifera. Next comes the colanterids. They have tentacles and bear nidoblast. Nidoblast is used for defense and capture of prey. They are mostly aquatic, sessile, and free-floating. Then comes the ctenophores. Ctenophores are marine animals with comb plates. So, ctenophores வந்துட்டு ஒரு ப்ராப்பர்ட்டி இருக்கும், bioluminescence property. It is property to emit light. So, it is the main characteristic feature of ctenophores. Then comes the platyhelminthes. Platyhelminthnave, it defines flat body. So, they have flat body and exhibit bilateral symmetry. The parasitic form show distinct suckers and hooks. So parasitic forms வந்து they have suckers and hooks. Husk helminthes are pseudocoelomate. and include parasitic as well as non-parasitic roundworms. So Ascalmenthes, they have roundworms. They are both parasitic or non-parasitic. They have pseudocoelamate. Pseudocoelamate na the body cavity is not covered by mesoderm. In either mesoderm it will be scattered into pouches. Then comes the annelids. Annelids are metamerically segmented. So annelids comes under the segmented organism with through coelom. Then comes the arthropods. Arthropods are the most abundant group of animals characteristic by presence of jointed appendages. So arthropods na like most of the insects, எல்லா insects வந்துட்டு it comes under arthropods. So they have jointed appendages. Then the mollusk. Mollusk have soft body surrounded by external calcareous shell. So molluskோட ஒரு definitive feature வந்துட்டு they have external calcareous shell. So mollusks தான் வந்துட்டு second largest phylum in the animal kingdom. First largest it comes to the arthropods. The body is covered with external skeleton made up of chitin. Next comes the echinoderms. Echinoderms possess a spiny skin. Their distinctive feature is the presence of water vascular system. So echinodermஸோட ஒரு definite feature வந்துட்டு water vascular system and they have a spiny skin. Arthropodsல they have jointed appendages. Mollusksல they have external calcareous shell and echinodermsல they have water vascular system. The hemichordates are a small group of worm-like marine animals. They have cylindrical body with proboscis, collar and trunk. So proboscis na it is head-like structure. Collar வந்துட்டு it is a body and trunk வந்துட்டு it is a tail-like structure. Then comes the phylum Chordata. Phylum Chordata includes animals which possess a notochord either throughout or during the early embryonic life. Other common features observed in the chordates are the dorsal hollow nerve cord and paired pharyngeal gill slits. Some of the vertebrates do not possess jaws. So the vertebrates which do not have jaws, they are known as agnatha. Whereas most of them possess jaws, they are known as gnatostomata. So the vertebrae which do not have jaws, they are known as agnatha. Which have jaws are known as gnatostomata. Agnatha is represented by the class Cyclostomata. So it is represented by a class Cyclostomata. So it is a class called Cyclostomata. They are the most primitive chordates and are ectoparasites on fishes. So they are primitive chordates and they are ectoparasite on fishes. Gnatostomata has two superclasses, Pisces and Tetrapoda. Then comes class Chondricytes and Osteocytes. Bear fins for locomotion and a group under Pisces. ஸோ நாத்தோஸ்டோமாட்டாவில் வந்து ரெண்டு கிளாஸ் இருக்குது, பிசஸ் அண்ட் டெட்ராபோடான்ட்டு. ஸோ பிசஸில் தேர் ஆர் அனதர் டூ கிளாஸ், கான்ட்ரிகைட்டிஸ் அண்ட் ஆஸ்டியோகைட்டிஸ். The chondricytes are fishes with cartilaginous endoskeleton and a marine. Classes Amphibia, Reptilia, Aves, Mammalia have two pairs of limbs and that is grouped under Tetrapoda. So chondricytes fishesல வந்து they have cartilaginous endoskeleton and osteichthisல வந்து they have bony endoskeleton. அப்புறம் Amphibia, Reptilia, Aves, Mammalia வந்து two pairs of limbs இருக்கனால they classified as Tetrapoda. The amphibians have adapted to live in both land and water. So amphibians can live in both land and water. Reptiles are characterized by the presence of dry and cornified skin. So reptilesல வந்து they have dry and cornified skin. Example வந்துட்டு snakes and crocodile and all. Limbs are absent in snakes. Fishes, amphibians and reptiles are poikothalmus. They are cold-blooded animals. Aves are warm-blooded animals with feathers on their bodies and four limbs modified into wings. So aves are birds. Birds are warm-blooded animals. They have feathers and four limbs are modified into wings for flying. And eye limbs are adapted for walking, swimming, perching, and clasping. The unique features of mammals are the presence of mammary glands and hairs on the skin. So mammals, like we compare the mammals. So நம்மளோட unique feature வந்து we have hair on the skin and mammary glands. They commonly exhibit viviparity. So viviparity na the baby develop inside the mother's womb. So you need to know the difference between chordates and non-chordates. So chordateல the notochord is present and in non-chordate notochord is not present. Chordatesல central nervous system is dorsal, Non-chordates it is ventral. Chordates pharynx is perforated by gill slits. Non-chordates gill slits are absent. So chordates heart is present ventral and non-chordates heart is present dorsal. Chordates post anal tail is present, but non-chordates it is absent. So you need to know this tabular column. So இதில் வந்துட்டு பர்டிகுலராக வந்துட்டு ஸோ எல்லா ஃபைலமோட ஆர்கனைசேஷன் சிமெட்ரி சீலோம் சிக்மெண்டேஷன் டைஜஸ்டிவ் சிஸ்டம் சர்க்குலேட்டரி சிஸ்டம்ஸ் எல்லாமே கொடுத்துருக்காங்க. ஸோ ஒரு லாஸ்ட் எல்லா சிஸ்டமும் படிச்சுட்டு யூ கேன் இதை பார்த்தோன்னா யூ கெட் டு நோ இதில் எது ப்ரெசன்ட் இதில் அது ஆப்சன்ட் இதில் இன்கம்ப்ளீட் இதில் கம்ப்ளீட் அந்த மாதிரி. ஸோ தட்ஸ் ஆல் ஃபார் அனிமல் கிங்டம். யூ நீட் டு நோ வாட் ஆர் ஆல் த ஃபைலம் அண்ட் தேர் யூனிக் கேரக்டரிஸ்டிக்ஸ். Then comes structural organization. Cells, tissues, organs, and organ systems split up the work in a way that ensures the survival of body as a whole and exhibit division of labor. So, our body is made up of cells. So, group of cells we call it as tissues. Though in the cells, tissues, organ system எல்லாமே வந்துட்டு நம்ம பாடியில் நடக்கிற ஒர்க் எல்லாம் வந்துட்டு divide. There will be division of labor. A tissue is defined as group of cells along with intercellular substance performing one or more function in the body. So tissue is defined as group of cells. It has intercellular substance. It performs one or more functions in the body. So a group of cell வந்துட்டு நம்ம பாடிக்குள்ள it performs some functions. Epithelia are sheet-like tissues lining the body surface and its cavities, ducts and tubes. Epithelia have one free surface facing the body fluid or the outside environment. So epithelia are the tissue, they line the body cavity and surface, ducts, tubes எல்லாமே. So there are different types of epithelia, squamous epithelium, columnar epithelium, cuboidal epithelium and all. Epithelia have free surface, like one surface faces the body fluid like blood and all, and another surface it faces the environment. The cells are structurally and functionally connected at junctions. So there are different types of junctions in our body like tight junction and gap junctions and all. The Indian bullfrog. So now we see about frog. So the Indian bullfrog, Rana tigrina, is a common frog found in India. Body is covered by skin. So the body of frog is covered by skin. Then mucus glands are present in the skin which is highly vascularized and help in respiration in water and on land. So it has mucus gland which help in respiration. Body is divisible into head and trunk. So body is divisible into head and trunk. A muscular tongue is present which is bilobed at the tip and it is used in capturing the prey. So it has bilobed tongue. The alimentary canal consists of esophagus, stomach, intestine, rectum, which open into cloaca. So cloaca is the excretory organ of frog. Like we have urethra, it has cloaca. The main digestive glands are liver and pancreas. So the digestive glands are liver and pancreas. It can respire in water through skin and through lungs on land. So frog is amphibian. In water, it respires through the skin and in land, it respires through the lungs. Circulatory system is closed with single circulation. So circulatory system is closed, the blood flow in the blood vessel. It is a single circulation. RBCs are nucleated as nucleated RBCs. Nervous system is organized into central, peripheral and autonomic nervous system. So it has central, peripheral and autonomic nervous system. The organs of urogenital system are kidneys and urogenital ducts. So it has kidneys and urogenital ducts which open into the cloaca. So cloaca are the excretory organ. The male reproductive organ is a pair of testes. Female reproductive organ is a pair of ovaries. So male reproductive organ is testes and female reproductive organ is ovaries. The female lays 2,500 to 3,000 ova at a time. The fertilization and development are external. The eggs hatch into tadpoles which metamorphose into dogs. So first the egg வந்துட்டு agitated form tadpole. Tadpoleக்கும் frogக்கும் வந்துட்டு like ரொம்ப difference இருக்கும். Then it metamorph. அது changes to frog. Now see about cockroach. The body of cockroach, Periplaneta americanus, covered by chitinous exoskeleton. So it has chitin. It is divided into head, thorax and abdomen. So head, thorax and abdomen. Segments bear jointed appendages. So cockroach is an insect. It comes under arthropoda. So arthropoda or unique character, they have jointed appendages. There are 3 segments of thorax, each bearing a pair of walking legs. So thorax there are 3 segments. Each segment bears a pair of walking legs. 2 pairs of wings are present. 1 pair each on second and third segment. There are 10 segments in abdomen. So abdomen there are 10 segments and thorax there are 3 segments. Alimentary canal is well developed with a mouth surrounded by mouth parts, a pharynx, esophagus, crop, gizzard, midgut, hindgut and anus. So crop is something which is used for storing food and gizzard is something which is used for grinding the food. Hepatic CK are present at the junction of foregut and midgut. So hepatic CK, it secretes digestive juices. Malphigian tubules are present at the junction of midgut and hindgut and help in excretion. So in cockroach, cloaca help in excretion. Here Malphigian tubules help in excretion. A pair of salivary gland is present near crop. The blood vascular system is open type. So the blood is open type. So the blood is not flowing in the blood vessel. Respiration takes place by network of tracheae. Tracheae opens outside with spiracles through which the respiration occurs. Nervous system is represented by segmentally arranged ganglia and ventral nerve cord. So here the nervous system is not well defined. It is arranged by ganglia and ventral nerve cord. A pair of testes is present at the 4th, 6th segment and ovary is 2nd and 6th segment. So you need to know the difference. Testes on the 4th and 6th segment, ovaries on the 2nd and 6th segment. Fertilization is internal. Female produces 9 to 10 ootheca bearing developing embryos. After rupturing of single ootheca, 16 egg ones called nymphs compound. In frogs, egg hatch we call it as tadpoles. Here we call it as nymph. So that's all about frog and cockroach. You need to know like how they respire, excrete, those things and all, and what are their definite characteristics and what are these different characteristics. Then comes the cell unit of life. So all organisms are made up of cells and aggregates of cells. So all organisms they are made up of cells and group of cells. Cells vary in their shape, size, activities or functions. So every cell differ in their shape, size, activities or function. Based on the presence or absence of membrane-bound nucleus and other organelles, cells and hence organisms can be named as eukaryotic and prokaryotic. எப்படி நம்ம வந்து eukaryotic and prokaryotic னு சொல்லுவோம்னா based on the presence and absence of membrane-bound nucleus and organelles. A typical eukaryotic cell consists of cell membrane, nucleus and cytoplasm. Or a typical eukaryotic cell, if we take cell of us, it has a typical eukaryotic cell, plant cell எடுத்துட்டோம்னா it has cell membrane, nucleus and cytoplasm. So plant cell have cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates transport of several molecules. So ஒரு plant cell எடுத்தோம்னா it has cell wall outside the cell membrane. But animal cell doesn't have a cell wall outside the cell membrane. So the plasma membrane is selectively permeable and facilitates transport of several molecules. So it helps in transport of molecules. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome and vacuoles. So ஒரு endomembrane systemல வந்துட்டு ஸோ எண்டோமெம்பரேன் சிஸ்டமில் வந்து 4 ஆர்கனல்ஸ் இருக்கும். எண்டோபிளாஸ்மிக் ரெட்டிகுலம், கோல்கி காம்ப்ளெக்ஸ், லைசோசோம் அண்ட் வேக்யூல்ஸ். ஸோ பிளாஸ்மா மெம்பரேனில் வந்துட்டு தெர் இஸ் அ மாடல் கால் ஃப்ளூயிட் மொசைக் மாடல். அந்த ஃப்ளூயிட் மொசைக் மாடல் என்ன பண்ணுன்னா அதில் வந்து லைக் வி காட் டு நோ த ஃப்ளூயிடிட்டி ஆஃப் த பிளாஸ்மா மெம்பரேன். All cell organelles perform different but specific functions. Centrosome and centriole form the basal body of cilia and flagella that facilitate locomotion. So centrosome and centrioles வந்துட்டு they form basal body of cilia and flagella and facilitate helps in locomotion. In animal cells, centrioles also form spindle apparatus during cell division. So animal cellsல வந்துட்டு they form spindle apparatus. So they during cell division. Nucleus contains nucleoli and chromatin network. It not only controls the activity of organelles but also play a major role in heredity. So nucleusல வந்துட்டு it has a nuclei and chromatin network. Nuclei it is ஒரு small like ball like structure. Nucleus பக்கத்துலயே இருக்கும் and chromatin network. இது வந்துட்டு வெறும் like எல்லா organellோட activity மட்டும் control பண்ணாது. They play a major role in heredity. Endoplasmic reticulum contains tubules or cisternae. So endoplasmic reticulum it has tubules or cisternae. There are two types of endoplasmic reticulum, rough and smooth. So based on the appearance, it is classified into rough and smooth. Rough endoplasmic reticulum it has ribosomes. Smooth endoplasmic reticulum ribosomes irukadu. ER helps in the transport of substances, synthesis of proteins, lipoproteins and glycogen. So rough endoplasmic reticulumல ribosomes இருக்கனால protein synthesisக்கு it will useful. Smooth endoplasmic reticulumல lipid synthesis also can take place. So endoplasmic reticulum வந்து மேஜரா எதுக்குன்னா transport of substances, so synthesis of proteins, lipoproteins and glycogen. The Golgi body is membranous organelle composed of flattened sacs. So Golgi bodiesல வந்துட்டு it have flattened sacs. The secretions of cell are packed in them and transported from the cell. So secretion and transport is done by Golgi body. Lysosomes are single membrane structures containing enzymes for digestion of all types of macromolecules. So lysosomesல வந்துட்டு it has different enzymes for digestion. So it works on acidic pH. It has different enzymes like proteases, carbohydrates, lipases, nucleases and all. Ribosomes are involved in protein synthesis. So ribosomes are non-membrane bound organelle. They are involved in protein synthesis. They occur freely in the cytoplasm, are associated with ER. So if it is associated with endoplasmic reticulum, it is called rough endoplasmic reticulum. Mitochondria are known as powerhouse of the cell because they help in oxidative phosphorylation and generation of ATP. They are bound by double membrane. The outer membrane is smooth and the inner membrane folds into several cristae. So endoplasmic reticulum it is cisternae. Mitochondria it is cristae. So mitochondria is a double membrane bound organelle. Outer membrane and inner membrane. Outer membrane is smooth and inner membrane has the folds. Plastids are pigment containing organelles found in plant cells only. So plastids are found in only plant. So plastidsல வந்து they are chloroplasts, leucoplasts and all. Chloroplastல தான் வந்து it has pigment and helps in photosynthesis. So here it is in plants it's chloroplast are responsible for trapping light energy essential for photosynthesis. The grana in the plastid is a site of light reaction and the stroma for dark reaction. So plastid light has grana and stroma. Grana for light reaction and then stroma for dark reaction. The green colored plastids are chloroplast which contain chlorophyll, whereas the other colored plastids are chromoplast which may contain pigments like carotene, xanthophyll. So chromoplastல like other pigments இருக்கும் like carotene and xanthophyll. Chloroplastல it has chlorophyll. The nucleus is enclosed by nuclear envelope, a double membrane structure with nuclear pores. So nucleus வந்துட்டு it is enclosed by a nuclear envelope. அது வந்து ஒரு double membrane structure. It has pores in between which is called nucleus pores. So எப்படி இருக்கும்னா it will be like this. So here the nucleus will be இதுதான் வந்துட்டு nuclear pore. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, the cell is structural and functional unit of life. So, cell is the structural and functional unit of life. ஸோ இந்த லெசன் பொறுத்த வரைக்கும் யூ நீட் டு நோ லைக் எது வந்து மெம்பரேன் பவுண்ட் ஆர்கனல் எது நான் மெம்பரேன் பவுண்ட் ஆர்கனல். ஸோ ரைபோசோம் இஸ் த நான் மெம்பரேன் பவுண்ட் ஆர்கனல். லைக் வாட் ஆர் த ஃபங்க்ஷன்ஸ் ஆஃப் ஈச் ஆர்கனல்ஸ் யூ ஷுட் நோ. So they will ask from this diagram like what is the size of each cell. So a typical eukaryotic cell size is 10 to 20 micrometer and bacteria has 1 to 2 micrometer and PPLO has 0.1 micrometer. Viruses has 0.02 to 0.2 micrometer. So they will ask from this question about the size and all. So that's all for cell unit of life. Then we go for biomolecules. Although there is bewildering diversity of living organisms, their chemical composition and the metabolic reaction appear to be remarkably similar. The elemental composition of living tissues and non-living matter appears also to be similar when analyzed qualitatively. So this is the comparison between the elements present in living and non-living matter. So you need to know like living எது வந்து ரொம்ப அதிகமாக ப்ரெசென்ட் ஆயிருக்கு எது வந்து ரொம்ப கம்மியாக ப்ரெசென்ட் ஆயிருக்குன்ட்டு you should know. However, a closer examination reveals that the relative abundance of carbon, hydrogen and oxygen is higher in living system when compared to inanimate matter. So the carbon, hydrogen, oxygen and all is very more in the living system than in the non-living system. The most abundant chemical in living organism is water. There are thousands of small molecular weight, less than 10,000 datals biomolecules. Amino acids, monosaccharide and disaccharide, sugars, fatty acids, glycerol, nucleotides, nucleosides, and nitrogen bases are some of the organic compounds seen in living organism. So, in living organism, we have amino acids, monosaccharide, disaccharides, nitrosides, nucleotides, nucleosides, nitrogen bases, and organic compounds. There are 20 types of amino acids and 5 types of nucleotides. So there are 20 types of amino acids. Amino acids are we can like further classify as essential amino acids and non-essential amino acids. So bookla vandu like they given 3 example. Acidic amino acid ku vandu glutamic acid. And then for basic they given lysine. And for neutral value. So you need to know these 3 examples. And there are 5 nucleotides. The 5 nucleotides are adenine, guanine, cytosine, uracil and thymine. So always adenine pairs with thymine and guanine pairs with cytosine. Fats and oils are glycerides in which fatty acids are esterified to glycerol. So fats and oils are glycerides. Then fatty acids are esterified to glycerol. Phospholipids contain, in addition, a phosphorylated nitrogenous compound. Only 3 types of macromolecules, proteins, nucleic acid, and polysaccharide are found in living system. So, living system there are 3 types of macromolecules. Macromolecules are large molecules, proteins, nucleic acids, and polysaccharides. Lipids, because of their association with membranes, separate in the macromolecular fraction. So lipids வந்து like plasma membrane lipids இருக்கும். So in association with the membranes, they separate in the macromolecular fraction. Biomacromolecules are polymers. So biomacromolecules are polymers. They are made up of building blocks which are different. Proteins are heteropolymers made up of amino acids. So ஒரு protein வந்து made up of different amino acids. Nucleic acids, RNA and DNA are composed of nucleotides. Biomacromolecules have a hierarchy of structures: primary, secondary, tertiary, and quaternary structure. So, proteins have 4 structures: primary, secondary, tertiary, and quaternary structure. So, primary it will be arranged in single line. Secondary structure as beta pleated sheets and alpha helix. Like this it will be. Then tertiary and quaternary. So further the secondary structures will undergo further complications and will form tertiary and quaternary structures. Nucleic acids serve as genetic material. Polysaccharides are components of cell wall in plants, fungi, and also of the exoskeleton of arthropods. So polysaccharides are components of cell wall in plants, fungi, and exoskeleton of arthropods. They also are storage forms of energy. Example, starch and glycogen. So polysaccharides also storage forms of energy. Example on the starch and glycogen. Proteins serve a variety of cellular functions. Many of them are enzymes. So proteins வந்துட்டு நிறைய proteins வந்துட்டு they are like enzymes. Some are antibodies, some are receptors, some are hormones, and some others are structural protein. So நிறைய வந்துட்டு it is like enzymes only. அப்புறம் like others are antibodies, receptors, hormones, and structural proteins. Collagen is the most abundant protein in animal world, and ribulose biphosphate carboxylase oxygenase is the most abundant protein in the whole biosphere. So in animal world வந்துட்டு it is collagen, and rubisco it is in whole biosphere. Enzymes are proteins which catalyze biochemical reaction in the cells. So enzymes are proteins, they catalyze the biochemical reaction. So they enhance the product formation. Ribozyme are nucleic acids with catalytic power. Proteinaceous enzymes exhibit substrate specificity, require optimum temperature and pH for maximal activity. So this enzyme has so specific characters like substrate specificity, it requires optimum temperature and pH. So on the particular temperature and pH, its activity will be more. They are denatured at high temperature. So proteins get denatured at high temperature, it get collapse. Enzymes lower activation energy of reaction and enhance greatly the rate of reaction. So enzyme வந்து activation energy வந்து reduce பண்ணிட்டு rate of reaction வந்துட்டு it increases. Nucleic acids carry hereditary information and are passed on from parental generation to progeny. So nucleic acid it has DNA. So it pass on information from parental generation to the progeny. So enzymes வந்துட்டு it has competitive inhibition and non-competitive inhibition. So competitive inhibition வந்துட்டு ரெண்டு என்சைமோட ஸ்ட்ரக்சர் வந்து சேமாக இருக்கும். ஸோ தே காம்படிட் ஃபார் த சேம் ஆக்டிவ் சைட். நான் காம்படிட்டிவ் இன்ஹிபிஷனில் வந்து there will be no competition. Then comes the cell cycle. So according to the cell theory, cells arise from pre-existing cells. So according to cell theory, ஒரு செல் arise from pre-existing cell. ஸோ ஒரு ஃபர்ஸ்ட் இருக்க ஒரு செல்லில் இருந்து தான் இன்னொரு செல் வருது அப்படின்னு சொல்கிறாங்க. The process which this occur is called cell division. So this process is called cell division. Any sexually reproducing organism starts its life cycle from a single-celled zygote. ஸோ ஒரு செக்ஷுவலி ரீப்ரொடியூசிங் ஆர்கனிசம் வந்து எப்படி அதை லைஃப் ஸ்டார்ட் பண்ணணும்னா சிங்கிள் செல்ட் ஜைகோட். ஸோ we know like male and female gamete fuse to form a zygote. Cell division does not stop with the formation of mature organism but continues throughout its life cycle. So cell division வந்துட்டு ஒரு mature organism பார்த்ததுமே வந்து it doesn't stop. Throughout our life it will continues. The stages through which a cell passes from one division to the next is called the cell cycle. So cell cycle is divided into two phases. Interphase, a period of preparation for cell division, and mitosis, M phase, the actual period of cell division. So cell cycle வந்து there are two phases, interphase and M phase. Interphase வந்துட்டு the cell will prepare itself for the cell division, and then M phase. M phase the mitotic phase where the actual cell division occurs. Interphase is further subdivided into G1, S and G2. So G1, S, and G2. G1 phase is the period when the cell grows and carries out normal metabolism. Most of the organelle duplication occurs during this phase. So G phase la most of the organelles get duplicate. S phase marks the phase of DNA replication and chromosome duplication. So S phase is very important where the DNA replicates. G2 phase is the period of cytoplasmic growth. So G2 phase le it occurs cytoplasmic growth. Mitosis is also divided into 4 stages, namely prophase, metaphase, anaphase and telophase. So M phase வந்துட்டு it is divided into 4 stages. Prophase, metaphase, anaphase and telophase. Chromosome condensation occurs during prophase. So prophase like it occurs condensation of chromosome. Simultaneously, the centrioles move to the opposite poles. Nuclear envelope and the nucleolus disappear and the spindle fibers start appearing. So prophase la என்னலாம் நடக்கும்னா there will be condensation of chromosome. ஸோ வந்து இந்த ரொம்ப வளைஞ்சு வளைஞ்சு இருக்க குரோமோசோம் இட் ஸ்டார்ட் டு கண்டென்ஸ். அதுக்கப்புறம் இந்த சென்ட்ரியோல்ஸ் ஆனால் மூவ் டு த ஆப்போசிட் சைட். இந்த சென்ட்ரியோல்ஸ்லாம் வந்துட்டு ஸ்பிண்டில் ஃபைபர்ஸ் ப்ரொடியூஸ் பண்ணும். இந்த சென்ட்ரியோல்ஸ் வந்து ஆப்போசிட் சைடுக்கு மூவ் ஆகிடும். அண்ட் தென் இந்த நியூக்ளியோலஸ் இதெல்லாமே நியூக்ளியர் என்வலப்லாம் வந்துட்டு டிஸப்பியர் ஆகிடும். மெட்டாஃபேஸ் இஸ் மார்க்ட் பை அலைன்மெண்ட் ஆஃப் குரோமோசோம் at the equatorial plate. ஸோ மெட்டாஃபேஸில் என்ன நடக்கும்னா குரோமோசோம் வந்து ஈக்குவட்டோரியல் பிளேட் லைக் சென்டர் பிளேட்டில் வந்து இட் கெட் அலைன். During anaphase, the centromeres divide and the chromatids start moving towards the opposite poles. So, anaphase la centromeres divide. Once the chromatids reach the two poles, the chromosomal elongation start, nucleolus and the nuclear membrane reappear. So, once, like after centromere divide and they went to the opposite poles, chromatids, chromosomal elongation, nucleolus, nuclear membrane எல்லாமே reappear ஆக ஸ்டார்ட் ஆகும். ஸோ இந்த ஸ்டேஜ் தான் we call it as telophase, reappearation ஆக. Nuclear division is then followed by cytoplasmic division and is carried, it is called cytokinesis. So after nuclear division, there will be cytoplasmic division. இதை வந்துட்டு we call it as cytokinesis. Mitosis, thus, it is the equational division in which the chromosome number of the parent is conserved in the daughter cell. So mitosis is the equational division. So parents la irukka same number of chromosome will come to the daughter cell. In contrast to mitosis, meiosis occurs in diploid cells, which are destined to form gametes. It is called reduction division. So mitosis is called equational division. Meiosis is called reduction division. Why? Since it reduces the chromosome number by half while making gametes, then in sexual reproduction, when the two gametes fuse, the chromosome number is restored to the value in the parent. So meiosis is known as reductional division because it divides to form gametes. Gametes are haploid, then it fuses again to form diploid. Meiosis is divided into two phases, meiosis 1 and meiosis 2. The first meiotic division is the homologous chromosome pair to form bivalents and undergo crossing over. So meiosis 1 there will be pairing of chromosome, crossing over and all will take place. Meiosis 1 has long prophase, which is divided further into 5 phases: leptotene, zygotene, pachytene, diplotene and diakinesis. So meiosis 1 prophase வந்துட்டு it is divided further into 5 stages: leptotene, zygotene, pachytene, diplotene and diakinesis. ஸோ லைக் இந்த ஒரு ஸ்டேஜஸில் வந்துட்டு ஒன்று ஒன்று நடக்கும். லைக் லிப்ரோடைனில் வந்துட்டு லைக் குரோமோசோம் கண்டென்சேஷன் வந்து நடந்துகிட்டே இருக்கும். தென் இன் ஜைகோடைனில் வந்துட்டு பேரிங் ஆஃப் குரோமோசோம் அக்கர்ஸ். பேரசிடைனில் வந்து தட் பைவேலண்ட் and all clearly they appears. And then in diplotain, the chiasma will be prominently present at the site of recombination. அந்த recombination எங்கே நடந்துச்சோ அந்த egg shaped chiasma மட்டும் பர்டிகுலராக தெரியும். Then in diakinesis, the termination of chiasma will occur. During metaphase 1, the bivalents arranged on the equatorial plate. So metaphase 1 அப்போ the bivalents and all arranged on the equatorial plate. This is followed by anaphase 1, the homologous chromosome move on to the opposite poles. So ரெண்டு ஒட்டி இருக்க குரோமோசோம் வந்து தனித்தனியாக ஒன்னோட move to the opposite poles in anaphase 1 with both the chromatids. Each pole receive half of the chromosome number of the parent cell. In telophase 1, the nuclear membrane and nucleolus reappear. So telophase 1 the nucleolus and nuclear membrane all reappear. Meiosis 2, it is similar to mitosis. So meiosis 2 is similar to the mitosis, prophase, metaphase, anaphase, telophase. So இப்போ ஒரு chromosome pole पे it gets divided and it further undergoes meiosis 2. It is very similar to the mitosis. During anaphase 2, the sister chromatids separate. Thus, at the end of meiosis, 4 haploid cells are formed. So you need to know what happened in G1, S and G2 and what happened in M phase, like mitotic phase. அப்புறம் meiosis 1 pro phaseல like இந்த ஸ்டெப்ல என்னென்ன நடக்கும்னு you should know. Then comes the breathing lesson. Cells utilize oxygen for metabolism and produce energy along with substance like carbon dioxide, which is harmful. So cell utilize oxygen for metabolism and produce energy along with substance like carbon dioxide, which is harmful. Animals have evolved different mechanisms for the transport of oxygen to the cell and for removal of carbon dioxide from there. So animalsல வந்து there is a ஒரு particular system for transport of oxygen to the cells and the removal of carbon dioxide. We have well-developed respiratory system comprising 2 lungs and associated with air passages to perform this function. So we have, like human beings, we have a respiratory system which has 2 lungs for air passage to perform this function. The first step in respiration is breathing, by which atmospheric air is taken in inspiration and the alveolar air is released out expiration. So first step in respiration is breathing. So breathing na enna inspiration and expiration. Inspiration the air we inspire, expiration the air we expel out. Exchange of O2 and CO2 between deoxygenated blood and alveoli. So exchange of O2 and CO2 is between deoxygenated blood and alveoli. Transport of these gases throughout the body by blood, exchange of O2 and CO2 between oxygenated blood and tissues, and utilization of O2 by the cell. Cellular respiration are the other steps involved. Inspiration and expiration are carried out by creating pressure gradients between the atmosphere and the alveoli with the help of specialized muscle intercostals and diaphragm. So there are 2 types of intercostal muscle, external intercostal and internal intercostal muscle. So there is a pressure gradient is created, so with the help of intercostal and diaphragm. Volume of air involved in these activities can be estimated with the help of spirometer and or of clinical significance. So there are different types of volume involved, like tidal volume, inspiratory reserve volume, expiratory reserve volume, then residual volume and all. Exchange of O2 and CO2 at the alveoli and tissues occur by diffusion. So exchange of O2 and CO2 at the alveoli, the site of exchange of gases, it is occurred by diffusion. Rate of diffusion is dependent on the partial pressure gradients of O2, PO2 and CO2, PCO2. The solubility as well as thickness of the diffusion surface. So diffusion depends on partial pressure of oxygen, partial pressure of CO2, the solubility, and the thickness of the diffusion surface. These factors in our body facilitate diffusion of oxygen from alveoli to the deoxygenated blood as well as from the oxygenated blood to the tissues. These factors are favorable for the diffusion of CO2 in the opposite direction from tissues to alveoli. So there is oxygen dissociation curve. So oxygen dissociation curve ஓட இது வந்து என்னன்னா high O2, less CO2, low H plus ions like acidic environment. So it favors for oxygen dissociation curve. இதோட அப்படியே opposite வந்து it favors for like CO2 ஓட dissociation curve. Oxygen is transported mainly as oxyhemoglobin. In the alveoli where PO2 is higher, O2 gets bound to the hemoglobin, which is easily dissociated at the tissues where PO2 is low. And PCO2 and H plus concentration are high. So oxygen, when it is transported at oxyhemoglobin, it gets bound with the hemoglobin to form oxyhemoglobin, and it is easily dissociated at the tissues where PO2 is low and PCO2 and H plus concentration are very high. Nearly 70% of carbon dioxide is transported as bicarbonate with the help of enzyme carbonic anhydrase. So 70% of carbohydrate are transported as in bicarbonate ions with the enzyme called carbonic anhydrase. 20 to 25% of carbon dioxide is carried by hemoglobin by carbamino hemoglobin. So for oxygen it is oxyhemoglobin, for carbon dioxide it is carbamino hemoglobin. In the tissues where PCO2 is high, it gets bound to blood, whereas in the alveoli where PCO2 is low and PO2 is high, it gets removed from the blood. So tissues la vandittu PCO2 is high, so it get bound to the blood. Alveoli la PCO2 is low, so it get removed. Respiratory rhythm is maintained by respiratory center in the medulla region of the brain. So respiratory rhythm. ஸோ இப்போ திடீர்னு வந்துட்டு இஃப் யூ கெட் ரொம்ப அதிகமாக யூ நீட் டு ப்ரீத், இல்லை அதிகமாக ப்ரீத் ஆகிறப்போ யூ நீட் டு கண்ட்ரோல் இதெல்லாம் வந்துட்டு இந்த ரெஸ்பிரேட்டரி சென்டர்னால இட் ப்ரெசன்ட் இன் த மெடுலா. இட் இஸ் அ ரீஜன் ஆஃப் பிரெயின். இட் இஸ் present in hind brain. Pneumotaxic center in the pons region of the brain and a chemosensitive area in the medulla can alter respiratory mechanism. So pneumotaxic center in pons and chemosensitive area in medulla, they alter the respiratory mechanisms. So you need to know the partial pressures present at different sites for O2 and CO2. So O2 it is more atmospheric air, and CO2 it is very less 0.3. So you need to know this, so you get to know where it diffuses more and where it diffuses less and all. So that's all for breathing. Then comes the blood. Vertebrates circulate blood, a fluid connective tissue in their body, to transport essential substance to the cell and carry waste substance from there. So blood is a fluid connective tissue. It helps in transportation of substance and removal of waste substances. Another fluid, lymph, is also used in transport of certain substances. So there is another fluid called lymph. So lymph has no pigment, blood has a pigment. Blood comprises of fluid matrix, plasma and formed elements. So blood has fluid matrix, plasma and formed elements. Red blood cells, erythrocytes, white blood cells, WBC or leukocytes, and platelets, thrombocytes constitute the formed elements. So the formed elements are RBC, WBC and platelets. Blood of humans are grouped into A, B, AB and O system based on the presence and absence of two surface antigens A, B on the RBC. So these antigen are all present on the RBC surface. Based on that, it is classified into A, B and O. It has antigen A it has antigen B, it has both the antigen, and O has no antigen. So A has antibody B, anti-B, and B has antibody A, anti-A. So AB has no antibody, so O has both the antibody. Another blood grouping is also done based on the presence or absence of another antigen called rhesus factor on the surface of RBC. So rhesus factor D antigen, is it present on the RBC surface or not? The space between cells in the tissue contain a fluid derived from blood is called tissue fluid. This is known as lymph. This fluid is called lymph and almost similar to blood, except for the protein content and the formed elements. So lymph is similar to blood but it doesn't have the protein content and the formed elements. All vertebrates and few invertebrates have closed circulatory system. So closed circulatory system, the blood flows in the blood vessel. Our circulatory system consists of muscular pumping organ, heart, a network of vessels and a fluid, blood. So heart has two atria and two ventricles. So heart has two atria, two ventricles, right atria, right ventricle, left atria and left ventricle. Cardiac musculature is auto-excitable. So heart is a auto-excitable organ. Sinoatrial node generates the maximum number of action potential per minute, 72, 75 per minute, and therefore it sets the pace of the activities of the heart. So sinoatrial node, SA node, there is something in the heart. It present in the right upper corner of right side of the heart. So it produces maximum action potential. So it is known as pacemaker of the heart. So it is called the pacemaker of the heart. The action potential causes the atria and then the ventricles to undergo contraction, systole, followed by relaxation, diastole. So இந்த ஆக்ஷன் பொட்டன்ஷியல் என்ன பண்ணுன்னா ஏடிஎம் அண்ட் வென்ட்ரிகல்ஸை வந்துட்டு கான்ட்ராக்ட் பண்ணுறதுக்கும் ரிலாக்ஸ் பண்ணுறதுக்கும் யூஸ் ஆகும். இட் இஸ் கால்ட் சிஸ்டோல் அண்ட் டயஸ்டோல். The systole forces the blood to move from atria to ventricles and to the pulmonary artery and the aorta. So systole என்ன நடக்கும்னா the blood move from atria to the ventricles, then to the pulmonary artery and the aorta, then to the rest of the body. The cardiac cycle is formed by sequential events in the heart which is cyclically repeated and is called cardiac cycle. So there are sequence of steps involved in this process. So இதுதான் வந்து we call it as cardiac cycle. A healthy person shows 72 such cycle per minute. So இதுதான் we call it as 72 beats per minute. So normal heart rate வந்து 72 beats per minute அப்படின்னு சொல்றது வந்து இதனால தான். About 70 ml of blood is pumped out of each ventricle during a cardiac cycle and it is called stroke or beat volume. So stroke volume na the blood pumped out of each ventricle during cardiac cycle. It is 70 ml. Volume of blood pumped out by each ventricle of heart per minute is called cardiac output and it is equal to the product of stroke volume and heart rate, approximately 5 liters. The electrical activity of heart can be recorded from the body surface by using electrocardiograph and the recording is called electrocardiogram, which is of clinical importance. So in the electrical activity of heart it can be recorded using electrocardiograph and the record பண்ண system பேர் we call it electrocardiogram, ECG. So you need to know the different elevations in the ECG like S wave, QRS wave and T wave and all you should know. We have complete double circulation. Therefore, two circulatory pathways, namely pulmonary and systemic, are present. So there are two circulatory pathways, pulmonary pathway and systemic pathways. So pulmonary pathways between lungs and the tissues, systemic pathway within the tissues. The pulmonary circulation starts by pumping of deoxygenated blood by right ventricle, which is carried to the lungs, where it is oxygenated and returned to the left atrium. So pulmonary circulation deoxygenated blood comes out of the right ventricle, it then carried to the lungs. Lungs it get converted to oxygenated, then it comes to the left atrium. Systemic circulation, pumping of oxygenated blood by left ventricle to aorta, which is carried to the body tissue, and the deoxygenated blood from there is collected by the veins and returned to the right atrium. So systemic circulationல வந்துட்டு left ventricle இருந்துட்டு the oxygenated blood வந்துட்டு it goes to the aorta, then it carried to the other body parts, then deoxygenated blood from there வந்து இது collected by the veins and it is returned to the right atrium. Though the heart is auto-excitable, its function can be moderated by neural and hormonal mechanism. Though the heart is auto-excitable, without anything it can function on its own. It also needs neural and some hormonal mechanism. So that's all for blood. Then excretory system. Many nitrogen-containing substance, ions, CO2, water, etc. that accumulate in the body have to be eliminated. Nature of nitrogenous waste form and excretion may vary among animals, mainly depending on the habitat, availability of water. So the nitrogen containing substance, ions, CO2, water, இதெல்லாமே வந்துட்டு harmful substance. It get accumulated in the body and இதெல்லாமே வந்துட்டு நம்ம பாடியை விட்டு eliminate பண்ணணும். So அதுக்கு we have a special system called excretory system. Nature of the nitrogenous waste form and this excretion வந்து very different animals. So இப்போ ஹியூமன்ஸ்க்கு ஒரு மாதிரி இருக்கும், ஃப்ராகுக்கு ஒரு மாதிரி இருக்கலாம், ஃபிஷ்ஷுக்கு ஒரு மாதிரி இருக்கலாம். So ammonia, urea, uric acid are the major nitrogenous waste. So ammonia, urea and uric acid are the major nitrogenous waste. So excretion of ammonia and all, we need large amount of water. So fish and all, like their major excretory product will be ammonia. Protonephridia, nephridia, malphizian tubules, green glands and the kidneys are the common excretory organs in the animals. So malphizian tubules are all excretory organ of cockroaches, green glands for prawns, kidneys for humans. So these are all common excretory organs. They not only eliminate nitrogenous waste but also help maintenance of ionic and acid balance in the body. These structures not only eliminate the waste, they help to maintain the ionic and acid balance, the sodium and potassium balance in our body. In humans, the excretory system consists of one pair of kidneys, a pair of ureter, a urinary bladder, and a urethra. So, human excretory system has pair of kidneys, pair of ureter, a urinary bladder, and a urethra. Each kidney has over million of tubular structures called nephron. So, each kidney has tubular structures called nephrons. Nephrons is the functional unit of kidney. So, nephron is the functional unit of kidney. It has two portions, glomerulus and renal tubule. Glomerulus is a tuft of capillaries formed from afferent arteriole, fine branches of renal artery. So glomerulus, it is formed by afferent arterioles. Afferent arterioles are the branches of renal artery. The renal tubule starts with double-walled Bowman's capsule and is further differentiated into proximal convoluted tubule PCT, Headley's loop, and distal convoluted tubule DCT. The DCT of many nephron join to a common and connecting duct, which ultimately opens into renal pelvis through the medullary pyramids. So the DCT join to form a common collecting duct, so which opens into the renal pelvis through the medullary pyramids. The Bowman's capsule encloses the glomerulus to form malphizian or renal corpuscle. So this is the glomerulus, Bowman's capsule, PCT, Henle's loop, DCT, collecting duct. This both together is known as renal corpus. Urine formation involves three main processes: filtration, reabsorption, and secretion. So, urine formation la three major steps irukku: filtration, reabsorption, and secretion. Filtration is non-selective process performed by glomerulus using the glomerular capillary blood pressure. About 1200 ml of blood is filtered by the glomerulus per minute to form 125 ml of filtrate in the Bowman's capsule per minute, GFR. So this is known as glomerular filtration rate. Amount of blood filtered by the glomerulus per minute. It is known as glomerular filtration rate. ஸோ எப்படி ஃபில்ட்ரேஷன் நடக்கும்னா லைக் இங்கே வந்து ஒரு பேஸ்மெண்ட் மெம்பரேன் இருக்கும். ஸோ பவுமன்ஸ் கேப்சூலில் வந்து தே ஹேவ் செல்ஸ் கால் போடோசைட்ஸ். அந்த போடோசைட்ஸ் 2 போடோசைட்டில் வந்து இருக்க கேப் எல்லாமே வந்துட்டு பிகாஸ் ஆஃப் போர்ஸ், அந்த போர்ஸ் வழியாக இட் கெட் ஃபில்ட்ரேட்டட். JGA, juxtaglomerular apparatus, a specialized portion of nephron, plays a significant role in the regulation of GFR. Nearly 99% reabsorption of filtrate takes place to differentiate part of the nephrons. So 99% of reabsorption of filtrate takes place as a different parts of the nephron. PCT is the major site of reabsorption and selective secretion. So PCT is a major reabsorption it takes place. Henle's loop primarily help to maintain osmolar gradient within the kidney interstitium. So PCT la major reabsorption. Henle's loop helps to maintain the osmolarity gradient. DCT and collecting duct allow extensive reabsorption of water and certain electrolytes. So DCT and collecting tubule water and electrolytes get reabsorbed, which help in osmoregulation. So H plus, K plus, NH3 ammonia could be secreted into filtrate by tubules to maintain ionic balance and a pH of body fluids. So collecting tubules also secrete H plus ions, K plus ions, ammonia to maintain the ionic balance of the body fluids. A Coucher current mechanism operates between two limbs of loop of Henle and with vasa recta. Vasa recta is a capillary parallel to the Henle's loop. So vasa recta is a capillary, it presents parallel to the Henle's loop. The filtrate gets concentrated as it moves down the descending limb, but it is diluted by the ascending limb. So when the fluid gets moved down in the Henle's loop, it first get diluted. So first it get concentrated by the descending limb, then it get diluted by the ascending limb. Electrolytes and urea are retained in the interstitium by this arrangement. DCT and collecting duct concentrate the filtrate about 4 times from 300 osmolarity to the 1200 osmolarity, an excellent mechanism for conservation of water. So the DCT and collecting duct it concentrate the filtrate for 4 times 300 osmolarity to the 1200 osmolarity. It helps in conservation of water. Urine is stored in the urinary bladder till voluntary signal from the CNS carried out its release through urethra. This is known as micturition. Skin, lungs, liver also assist in excretion. Like by sweat, waste products comes out is by skin. Carbon dioxide comes out through lungs. So these are also a form of excretion. So this is a representation of counter current mechanism. Like it is for concentration of urine. So that's all for excretion. So in excretory system, you also need to know about kidney transplant and dialysis. Then comes the locomotion. Movement is an essential feature of all living beings. Protoplasmic streaming, ciliary movements of fins, limbs, wings, or some forms exhibited by animals. A voluntary movement which causes the animal to change its place is called locomotion. So movement is an essential feature of living being. Like protoplasmic streaming, it is done in amoebas, ciliary movements of fins, ஃபிஷ்ஷோட ஃபிட்ஸ் மூவ் ஆகுறது, லிம்ஸு, விங்ஸு இது எல்லாமே மூமெண்ட்ஸ். பட் ஒரு வாலண்ட்ரி மூவ்மெண்ட் விச் காஸ் த அனிமல் டு சேஞ்ச் இட்ஸ் பிளேஸ் அதுதான் வந்து லோகோமோஷன். ஸோ சும்மா வாலண்ட்ரி மூவ்மெண்ட் விச் டசன்ட் காஸ் சேஞ்ச் ஆஃப் பிளேஸ் வந்து டிஃபைன் அஸ் மூவ்மெண்ட். பட் அ மூவ்மெண்ட் விச் காஸ் அ சேஞ்ச் இன் பிளேஸ் வந்துட்டு வி கால் இட் அஸ் லோகோமோஷன். Animals move generally in search of food, shelter, mate, breeding, ground, better climate, or to protect themselves. So why animals need locomotion and all? They need locomotion for search of food, shelter, for mating, for breeding, for climate, to protect themselves and all. The cells of the human body exhibit amoeboid, ciliary, and muscular movements. So cells of human body has amoeboid movement, ciliary movement, and muscular movements. Locomotion and many other movements require coordinated muscular activities. Three types of muscle are present in our body. Skeletal. So there are three types of muscle present in our body: skeletal muscle, smooth muscle, and cardiac muscle. The skeletal muscles are attached to skeletal elements. They appear striated. So it has more striations and are voluntary in nature. So these are voluntary. It has more striations. It is multinucleate. The nucleus present at the periphery. Visceral muscles present the inner walls of visceral organs are not striated, involuntary. So visceral muscles are smooth muscles. So it is present in the inner walls of visceral organs. They are not striated and they are involuntary. Cardiac muscles are the muscles of heart. They have striation, branched, and involuntary. So then comes the cardiac muscles. It is the specialized muscles of heart. They have striations, they are branched, and they are involuntary. Muscles possess excitability, contractibility, extensibility, and elasticity. So these are the properties of muscle: excitability, contractibility, extensibility, and elasticity. Muscle fibers is the anatomical unit of muscle. So muscle fiber is a basic unit of a muscle. Each muscle fiber has very parallelly arranged myofibrils. So இதுதான் வந்து ஒரு muscle fiber. இதில் வந்து they have parallelly arranged things called myofibril. Each myofibril contain serially arranged unit called sarcomere. So first ஒரு muscle fiber. Muscle fiberல it has myofibril. Myofibrilல it has units called sarcomere. Sarcomere is the functional units. Each sarcomere has central A band made of thick myosin filaments and two of I bands made of thin actin filament on either side of it marked by Z line. So A band is made up of thick myosin filament and I band is made up of thin actin filaments. Actin and myosin are polymerized proteins with contractibility. The active sites for myosin on resting actin filament are masked by protein troponin. So the active sites of myosin are present in the uh, actin filament. So it is masked by a protein called troponin. Myosin head contain ATPase and has ATP binding site and active site for actin. So myosin headல வந்து it has enzyme ATPase and ATP binding site and active sites for actin. A motor neuron carries signal to the muscle fiber which generate an action potential in it. This causes the release of calcium from the sarcoplasmic reticulum. It refers to the cytoplasm of the sarcomere. It is a sarcoplasmic reticulum. Calcium activates actin, which binds to myosin head to form a cross bridge. These cross bridge pull the actin filaments, causing them to slide over the myosin filament and thereby causing contraction. Calcium are then returned to the sarcoplasmic reticulum, which inactivate the actin. Cross bridges are broken and muscles get relaxed. So this is the process of contraction and relaxation. You need to know this diagram. So first ஒரு motor neuron வந்துட்டு muscle fiberக்கு வந்து it gives a signal. So அந்த signal வந்ததுமே there will be release of calcium ions from the sarcoplasmic reticulum. இந்த calcium ions வந்து it activates the actin. So இந்த actin என்ன பண்ணணும்? It get bind to the myosin head. Myosin headல வந்து there will be active site for actin. So it get bind to that to form a cross bridge. So இதுதான் வந்துட்டு cross bridge. So in the cross bridge வந்துட்டு pull the actin filaments, causing them to slide over the myosin filament. So it get pulled. So thereby it causes contraction. Then the calcium gets returned to the sarcoplasmic reticulum. Calcium level decrease ஆனதுமே it inactivate the actin. So in the cross bridges broke ஆயிடும். So it causes relaxation of muscle. Repeated stimulation of muscle leads to fatigue. ஸோ ரிப்பீட்டட் ஸ்டிமுலேஷன் தான் லைக் சும்மா நம்ம வந்துட்டு கான்ட்ராக்ஷன் ரிலாக்ஷன் பண்ணிகிட்டே இருக்கிறது the muscle get tired. Muscles are classified as red and white fibers based on primarily out of red colored myoglobin pigment present. Bones and cartilage constitute our skeletal system. So bones and cartilage constitute our skeletal system. The skeletal system is divisible into axial and appendicular. Skull, vertebral column, ribs and sternum constitute the axial skeleton. So the vertebrae are cervical, thoracic, lumbar, sacral and coccygeal. And then ribs, there are like 12 pairs of ribs and the sternum. Limb bones and girdles form the appendicular skeleton. So there are two types of girdles, pelvic girdle and pectoral girdle. Three types of joints are formed between bones or between bone and cartilage. So there are fibrous, cartilaginous and synovial joints. So cartilaginous joints are between the bone and the cartilage. Synovial joints include ball and socket joint, hinge joint and all. Synovial joint allow considerable movements and therefore plays a significant role in locomotion. So synovial joints play a role in locomotion. So you need to know this contraction, relaxation, coupling theory, and then the three types of muscle, and then about the bones, like what are the vertebrae and about the girdles and all. Then comes the neural coordination. The neural system coordinates and integrates function as well as metabolic and homeostatic activities of all the organs. So neural system is the basic control and coordinating system in our body. It controls, coordinates and integrates the function of metabolic and homeostatic activities of all the organs. Neurons are the functional unit of neural system. They are excitable cells due to differential concentration gradient of ions across the membrane. So neurons are the functional unit of neural system. So these are the neurons. Sorry. So this is a neuron. The neuron has nucleus. These are known as dendrites. It has some granule-like substance called Nielsen's granule. It helps in protein synthesis, and this is the axon. This is the myelin sheet. The cell present is a swan cell. Axonal end and the synaptic knob. So the places where the myelin sheet are absent in the axon is known as nodes of Ranvier. The electrical potential difference across the resting neural membrane is called resting potential. So the electric potential difference when the membrane is at rest is known as resting potential. The nerve impulse is conducted along the axon membrane in the form of way of depolarization and repolarization. So depolarization na excitation of the neuron. Repolarization na relaxation of the neuron. A synapse is formed by the membrane of presynaptic neuron and the postsynaptic neuron, which may or may not be separated by a gap called synaptic cleft. ஸோ இப்போ இதுதான் வந்து ஒரு சினாப்டிக் நாப்னா இது வந்து ப்ரீ சினாப்டிக் நியூரானாக இருக்கும். இங்கே டென்ட்ரைட்ஸ் will act as a post synaptic neuron. இன்னொரு இருக்க கிராப் will be synaptic cleft. Chemicals involved in the transmission of impulse and the chemical synapses are called neurotransmitters. So there are two types of impulses, electrical impulse and chemical impulse. Electrical impulse are very fast. Chemical impulse, there will be involvement of neurotransmitters. So major neurotransmitter it is acetylcholine. Human neural system consists of two parts. Central neural system, the peripheral neural system. So central neural system, it has a brain and the spinal cord. The CNS consists of brain and spinal cord. Okay, the brain can be divided into three major parts: forebrain, midbrain, and hindbrain. So brain is divided into three parts: forebrain, midbrain, and hindbrain. So forebrain is present in the front, midbrain in the middle, hindbrain is present behind. The forebrain consists of cerebrum, thalamus, and hypothalamus. So forebrain it has three parts: cerebrum, cerebellum, and hypothalamus. The cerebrum is longitudinally divided into two halves that are connected by the corpus callosum. So cerebrum is divided into two halves. The two halves is connected by a structure called corpus callosum. A very important part of forebrain is hypothalamus. So hypothalamus is very important because it has major centers in it. It controls body temperature, eating, drinking, and all. Inner parts of cerebral hemisphere and a group of associated deep structures form a complex structure called limbic system. So inner groups of cerebral hemisphere constitute a structure called limbic system, which is concerned with olfaction, autonomic response, regulation of sexual behavior, expression of emotional reactions, and motivation. So the limbic system controls the sexual behavior, expression, emotional reactions, and the motivation. The midbrain receives and integrates visual, tactile, and auditory inputs. Hindbrain comprises pons, cerebellum, and medulla. So the hindbrain has pons, cerebellum, and medulla. The cerebellum integrates information derived from the semicircular canals of the ear and the auditory system. So the cerebellum it integrates the information it receives from the semicircular canal of the ear and the auditory system. The medulla contains centers which control respiration, cardiovascular reflex, and gastric secretions. Pons constitute fiber tracts that interconnect different regions of brain. So pons has a tract that connect the different regions of brain. So medulla has the centers for respiratory center and then the cardiovascular reflex center. Then comes the endocrine. There are special chemicals which act as hormones and provide chemical coordination, integration, and regulation in the human body. So there are chemical structures present in the body which provide chemical coordination, integration, and regulation. So these are known as hormones. These hormones regulate metabolism, growth, and development of our organs. The endocrine glands are certain cells. The endocrine system is composed of hypothalamus, pituitary, and pineal, thyroid, adrenal, pancreas, parathyroid, thymus, and gonads. Gonads include testes and ovary. So, the endocrine gland is composed of hypothalamus, pituitary, pineal, thyroid, adrenal, pancreas, parathyroid, thymus, and gonads. In addition to this, some other organs like gastrointestinal tract, kidney, heart also produce hormones. The pituitary gland is divided into three major parts, which is called pars distalis, pars intermedia, and pars nervosa. Pars distalis produce six trophic hormones. So first we start with pituitary gland. Pituitary gland has three parts: pars distalis, pars intermedia, and pars nervosa. This pars distalis and pars intermedia will work coordinately only, and pars nervosa is a posterior pituitary and pars distalis is a anterior pituitary. So anterior pituitary produces six trophic hormones. Pars intermedia secretes only one hormone. So hormones of pituitary are growth hormone, thyroid stimulating hormone, follicular stimulating hormone, melanocyte stimulating hormone. These are all the hormones of pituitary. While pars nervosa, posterior pituitary is also known as neurohypophysis. It secretes two hormones. Actually, it doesn't secrete two hormones. Oxytocin and then vasopressin. These are secreted by hypothalamus. This both the hormones get stored in the posterior pituitary. The pituitary hormones regulate the growth and development of somatic tissues and activities of peripheral endocrine glands. So they regulate growth and development of somatic tissues. Pineal gland secrete melatonin, which plays a very important role in regulation of 24-hour diurnal rhythm of our body, rhythm of sleep and state of being awake, body temperature, etc. So pineal gland secrete melatonin. Melatonin helps in rhythm of sleep. The thyroid gland hormones play an important role in the regulation of basal metabolic rate, development and maturation of central neural system, erythropoiesis, metabolism of carbohydrates, proteins and fats, menstrual cycle. Another thyroid hormone, thyrocalcitonin, regulates calcium levels in our body. So thyroid hormone is a very important hormone. So thyroid hormone plays like variety of important role. It regulates basal metabolic rate, maturation of a central neural system, erythropoiesis, metabolism of carbohydrates, proteins and fats. It regulates the menstrual cycle. So there is another thyroid hormone called thyrocalcitonin. It regulates the calcium levels. The parathyroid gland secrete parathyroid hormone, which increases the calcium levels and plays a major role in calcium homeostasis. So there are parathyroid gland, it present behind the thyroid gland. It also plays an important role in calcium homeostasis by increasing the blood calcium levels. The thymus gland secrete thymocytes, which plays a major role in the differentiation of T lymphocytes, which provides cell-mediated immunity. In addition, thymocytes also increase the production of antibodies to provide humoral immunity. Okay, then comes the thymus gland. So thymus gland secrete thymocytes. It plays an important role in differentiation of T lymphocytes. So the bone marrow produce the B cells and T cells. It get differentiated in the thymus. So when the age increases, the thymus get degenerated. The adrenal gland is composed of centrally located adrenal medulla and the outer adrenal cortex. So next one is the adrenal gland. So adrenal gland it has in the periphery, it has adrenal cortex, and it has center, it has adrenal medulla. The adrenal medulla secrete epinephrine and norepinephrine. These hormones increase alertness, pupillary dilation, piloerection, sweating, heartbeat, strength of heart contraction, rate of respiration, glycogenolysis, lipolysis, and proteolysis. So adrenal medulla secretes epinephrine and norepinephrine. These both are the hormones of fight and flight. They increase alertness, pupillary dilation, piloerection, etc., etc., those kind of things. The adrenal cortex secretes glucocorticoids and mineral corticoids. Glucocorticoids stimulate gluconeogenesis, lipolysis, proteolysis, erythropoiesis, cardiovascular system, blood pressure, and glomerular filtration rate. It inhibits the inflammatory reaction by suppressing the immune response. So glucocorticoids inhibit inflammatory reaction by suppressing immune response. Mineral corticoids regulate water and electrolyte contents of the body. So glucocorticoids are the gluconeogenesis, lipolysis, proteolysis, blood pressure, glomerular filtration. But mineral corticoids for water and electrolyte balance. The endocrine pancreas secrete glucagon and insulin. So pancreas there is islet of Langerhans. It has B cells. So it secretes glucagon and insulin. Glucagon stimulates glucogenolysis and gluconeogenesis, resulting in hyperglycemia. So glucagon is a hyperglycemic hormone. Insulin stimulates cellular glucose uptake and utilization of glucogenesis, resulting in hypoglycemia. So insulin is a hypoglycemic hormone. Insulin deficiency and or insulin resistant result in disease called diabetes mellitus. So if there is insulin deficiency or resistance to the insulin, it causes diabetes mellitus. Diabetes insipidus is caused by vasopressin. The testis secretes androgen, which stimulate the development, maturation, and function of male accessory sex organs. appearance of male secondary sex characters, spermatogenesis, male sexual behavior, anabolic pathways, and erythropoiesis. So testes secrete hormone called androgens, testosterone. It helps in development maturation of male accessory sex organ, secondary sexual characters, erythropoiesis, male sexual behavior, and spermatogenesis. Ovaries secrete estrogen and progesterone. Estrogen stimulate the growth and development of female accessory sex organs and secondary sex characters. Progesterone plays a major role in maintenance of pregnancy as well as mammary gland development and lactation. So estrogen is helpful in, for the development of secondary sexual characters, and progesterone plays a major role in maintenance of pregnancy and in development of mammary gland and helps in lactation. The atrial wall of heart produces an atrial natriuretic factor ANF. It decreases the blood pressure. So atrial wall of heart, it produces ANF. It decreases the blood pressure. Kidney produce erythropoietin, which stimulate the process of erythropoiesis. The GIT secretes gastrin, secreted cholecystokinin, and gastric inhibitory peptide. So GIT also secretes various hormones called gastrin, secreted cholecystokinin, and gastric inhibitory peptide. These hormones regulate the secretion of digestive juices and help in digestion. So these GIT hormones are all, it regulates the secretion of digestive juices and helps in digestion. So these are all for hormones. So in for hormones, there are like two pathways, like how they act on the specific organ. If it is a peptide hormone like thyroid and all, they directly act on the receptor. They act on the hormone receptor. But other hormones like steroid hormones, the sex hormones are all, they do not directly act on the receptor. Instead, they produce secondary messengers to act on the particular organ. So that's all. So 7:40 to 7:50. Let's take a break. Human reproduction. Humans are sexually reproducing and viviparous. So humans reproduce by sexual reproduction. The male reproductive system is composed of pair of testes, the male sex accessory ducts, and accessory glands and external genitalia. So the male reproductive system has pair of testes, male sex accessory ducts. So the accessory ducts includes retate testes, vas efferentia, vas deferens, and epididymis, and the accessory gland. Accessory glands are prostate gland, bulbourethral gland, and seminal vesicles. And as the external genitalia, the external genitalia is the penis. Each testis has 250 compartments called testicular lobules, and each lobule contains 1 to 3 highly coiled seminiferous tubules. So, one testilla there is 250 compartments. They are called testicular lobules. And the 250 compartments they have like 1 to 3 highly coiled seminiferous tubules. Each seminiferous tubule is lined by spermatogonia and Sertoli cells. So, seminiferous tubules is lined by spermatogonia and Sertoli cells. The spermatogonia undergo meiotic division leading to sperm formation, while Sertoli cells provide nutrition to the dividing germ cells. Though the spermatogonia it undergo division to form sperm. In the Sertoli cells, it is called nerve cells. It produces nutrition to the dividing sperm germ cells. The Leydig cell outside the seminiferous tubule synthesize and secrete testicular hormone called androgens. So outside the seminiferous tubules, it is lined by Leydig cells. The Leydig cells synthesize and secrete hormone called androgens, testosterone. The male external genitalia is called penis. So the penis is covered by foreskin at the tip of it, glans penis. The female reproductive system consists of pair of ovaries, a pair of oviducts. Oviducts are called as fallopian tubes, a uterus, a vagina, and external genitalia. A pair of mammary glands. So external genitalia includes pons pubis, clitoris, hymen, labia majora, and labia minora. The ovaries produce female gamete ovum and some steroid hormones, ovarian hormones. So the ovaries produce a female gamete egg ovum and some steroid hormone, ovarian hormones, estrogen and progesterone. Ovarian follicles in different stage of development are embedded in the stroma. The oviducts, uterus and vagina are female accessory ducts. So female accessory ducts includes oviducts, uterus and vagina. The uterus has three layers, namely perimetrium, myometrium and endometrium. So uterus has the three layers, perimetrium, myometrium and endometrium. Perimetrium is the outer layer. Myometrium is the middle muscular layer. It is made up of smooth muscles. And endometrium is the inner layer. The female external genitalia includes pons pubis, labia majora, labia minora, hymen and clitoris. The mammary gland are one of the female secondary sexual characters. So mammary gland is the one of the female secondary sexual characters. So the mammary gland is has 15 to 20 mammary lobes. It has cluster of cells. They group together to form a alveoli. Alveoli secretes milk. Alveoli cluster together to form mammary tubule. Then mammary duct. Then mammary ampulla. Then comes the lactiferous duct, through which the milk is ejected out. Spermatogenesis results in the formation of sperms that are transported by male sex accessory ducts. So spermatogenesis is a process of formation of sperm. It are transported by the male accessory ducts. Vast difference in pre-diversity testes are helpful in transportation of sperm. A normal human sperm is composed of head, neck, middle piece, and the tail. So a sperm has a head, neck, middle piece, and the tail. So it has a egg, neck, middle piece, and the tail. So the middle piece has the mitochondria, neck has centrioles. So the head has a part called acrosome. The process of formation of mature female gamete is called oogenesis. So formation of sperm is called spermatogenesis, and formation of female gamete is known as oogenesis. The reproductive cycle of female primates is called menstrual cycle. Menstrual cycle starts only after attaining sexual maturation, after puberty. So after attaining puberty, menstrual cycle starts. During ovulation, only one ovum is released per menstrual cycle. The cyclic changes in the ovary and the uterus during menstrual cycle are induced by changes in the levels of pituitary and ovarian hormones. The cyclic changes in the ovary and uterus during menstrual cycle are induced by the levels of pituitary and ovarian hormones. So the ovarian hormones includes estrogen and progesterone, and pituitary hormones LH and FSH. After coitus, sperms are transported to the ampulla. Ampulla is a part of fallopian tube where the sperm fertilize the ovum, leading to formation of diploid zygote. The presence of X or Y chromosome in the sperm determines the sex of embryo. So the sex of embryo is determined by the father. The zygote undergoes repeated mitotic division to form a blastocyst. So the zygotes undergo multiple mitotic division to form a blastocyst. Blastocyst has blastomeres, the cells of the zygote. So when the embryo is at 8 to 14 cell stage, it is known as morula. Which is the morula gets implanted in the uterus, resulting in pregnancy. After 9 months of pregnancy, the fully developed fetus is ready for delivery. The process of childbirth is called parturition, which is induced by complex neuroendocrine mechanism involving cortisol, estrogen, and oxytocin. So the fully developed fetus, when it's ready for delivery, it is known as parturition. It is induced by hormones called estrogen, oxytocin, and cortisol. Mammary glands differentiate during pregnancy and secrete milk after childbirth. So milk is secreted after the childbirth. The newborn baby is fed milk by the mother. Lactation is known lactation during the initial few months of growth. It has antibodies IgA. Cholesterol. So this is the human reproduction. This is spermatogenesis and oogenesis. So the process of differentiation from spermatids to spermatozoa is known as spermiogenesis. Release of sperm is known as spermiation. Then this is oogenesis. So this is how a seminiferous tubule will be there. So it has primary spermatocyte, secondary spermatocyte, then it has Sertoli cells which produce nutrition to the developing sperm. This is the menstrual cycle. So during the ovulation phase, the LH will be at its high level, which induces ovulation. The progesterone will be high at the luteal phase. So if the ovum get fertilized, there will be formation of zygote. For that, progesterone is important. If there is no pregnancy, the progesterone levels get low. Reproductive health. Reproductive health refers to total well-being in all aspects of reproduction: physical, emotional, behavioral, and social. So reproductive health collectively, it includes physical, emotional, behavioral, and social aspects. Our nation was the first nation in the world to initiate various action plans at national level towards attaining a reproductively healthy society. So India was the first nation to bring out various actions. Counseling and creating awareness among people about reproductive organs, adolescents, and associated changes, safe and hygienic sexual practices, sexually transmitted infection, including AIDS, is a primary step towards reproductive health. So, like educating people about the adolescents, about the reproductive organ, about the sexually transmitted diseases, hygiene, sexual practice is the first step they brought towards the reproductive health. Providing medical facilities and care to the problems like menstrual irregularities, pregnancy related aspects, delivery, medical termination of pregnancy, sexually transmitted infection, birth control, infertility, postnatal child and maternal management is another important aspect of reproductive and child healthcare management. So, reproductive and child healthcare management programs mainly focuses on the MTPs, STIs, birth control, infertility, postnatal child and maternal management. An overall improvement in reproductive health has taken place in our country as indicated by reduced maternal and infant mortality rates, early detection and cure of STIs, assistance to infertile couples, etc. Improved health facilities and better living conditions promoted an explosive growth of population. Such a growth necessitated intense propagation of contraceptive methods. So let's see various contraceptive methods. Various contraceptive options are available now, such as natural. So the natural method involves like withdrawal methods. The male partner withdraws the penis before the ejaculation happens. Then the traditional method, then the barrier method. Barrier method includes condoms, IUDs. There are IUDs are intrauterine devices. There are like non-medicated IUDs called Lippis loop and copper producing IODs, CUD, CU7 or some examples. They produce copper ions which suppresses sperm motility. There are pills, injectables, implants, surgical methods. Surgical methods includes vasectomy and tubectomy. In vasectomy, the vas deferens is cut, and in tubectomy, the fallopian tube is cut and tied. They are like permanent methods. So, an ideal contraceptive should be user friendly, easily available. It is effective, no or least side effect. It should be reversible. These are the effective ideal contraceptive. Though contraceptives are not regular requirements for reproductive health, one is forced to use them to avoid pregnancy or to delay or space pregnancy. So delay or space pregnancy, we can use IUDs and what. So medical termination of pregnancy is legalized in our country. So MTP is known as abortion. It is legalized in our country in 1971. MTP is generally performed to get rid of unwanted pregnancy due to rapes, casual relationship, etc. As also in cases where the continuation of pregnancy could be harmful or even fatal, either to the fetus or the mother or both. So NTP is usually done to get rid of unwanted pregnancy due to rapes or like casual relationship. If like continuation of the pregnancy can cause any damage to the both fetus and the mother or only to the mother or fetus, MTP is performed. Infections are diseases transmitted through sexual intercourse are called sexually transmitted diseases, STIs. Pelvic inflammatory diseases, stillbirth, infertility, or some complications of them. So, complication of sexually transmitted diseases are stillbirth, infertility, and pelvic inflammatory diseases. Some sexually transmitted diseases are AIDS, herpes, hepatitis, gonorrhea, syphilis, and all. Early detection facilitate better cure of these diseases. Avoiding sexual intercourse with unknown or multiple partners, use of condoms during coitus, or some simple precautions to avoid contracting STIs. So in MTPs, like amniocentesis, like the amniotic fluid from the womb is taken out to check if the gender of the baby is like female or male. During that time, MTPs are not, it is not done. Inability to conceive or produce children even after 2 years of unprotected sexual cohabitation is called infertility. Various methods are now available for such couples. In vitro fertilization (IVF), followed by transfer of embryo into female genital tract, is one such method and is common as test tube baby program. If the male partner is infertile, we can use artificial insemination like that and all. And IVF uh, is one of the commonest methods. Surrogacy is also done nowadays. Molecular basis of inheritance. Nucleic acids are long polymers of nucleotides. So nucleic acids are long polymers of, so nucleotide is formed by a number of nucleotides. While DNA stores genetic information, RNA mostly helps in transfer and expression of the information. So DNA stores the genetic information, RNA transfers it. Though DNA and RNA both function as genetic material, but DNA being chemically and structurally more stable. So DNA is more stable than the RNA, it is better genetic material. RNA is the first to evolve and DNA was derived from RNA. The hallmark of the double-stranded helical structure of DNA is the hydrogen bonding between the bases from opposite strands. The rule is that adenine pairs with thymine through 2 hydrogen bonds and guanine with cytosine through 3 hydrogen bonds. So adenine always pair with thymine, guanine with cytosine. So this confers for the stability of the DNA. This makes one strand complementary to other. The DNA replicates semi-conservatively. So the DNA replication occurs semi-conservatively. So during the replication, there is an enzyme used called DNA-dependent DNA polymerase and DNA ligase and all. So this is how the DNA replicates. So it is a semi-conservative method. The strand divides and each strand acts as a template strand for the continuous. In 5' to 3', it is continuous synthesis, and 3' to 5', it is discontinuous synthesis. These fragments are called Okazaki fragments. These get attached by DNA ligase. A segment of DNA that codes for RNA, in a simplistic term, can be referred to as gene. A DNA that codes for RNA is referred to as gene. During transcription also, one of the strand of DNA acts as template to direct the synthesis of complementary RNA. So during transcription, after replication, during transcription, one of the strand acts as the template strand. The template strand is 3 end to 5 end. In bacteria, the transcribed mRNA is functional, hence can directly be translated. In eukaryotes, the gene is split. The coding sequence exons are interrupted by non-coding sequence introns. So this intron should be removed and exons should be joined to form a functional RNA. This process is by splicing. So this is a transcription in bacteria. So the promoter factor is a sigma factor. It starts with the initiation of the transcription. The RNA is transcribed and it binds with rho factor, which is a termination factor, and causes termination. But in eukaryotes, the introns are interrupted in between. So these introns should be removed. So this process is called splicing, and the exons are like attached together. And capping is happen at the 5' end by adding methyl guanosine phosphate, and tailing is done at the 3' end by adenyl residues. So this is about transcription. The messenger RNA contains the base sequence that the rendered combination of 3 to make triplet genetic code to code for an amino acid. The genetic code read again on the principle of complementary by tRNA that act as adapter molecule. So the tRNA as The tRNA has three codes. Its complementary code. It attaches a complementary code to form a protein. There are specific tRNA for every amino acid. The tRNA binds to specific amino acid at one end and passes through H bonding, which codes an mRNA through its anticodons. The tRNA has anticodons on the mRNA. It binds by a hydrogen bonding and translates to a protein. The site of translation protein synthesis is ribosomes. So the translation takes place in the ribosomes, which binds to mRNA and provides platform for joining of amino acids. One of the rRNA acts as catalyst for peptide bond formation. So for translation, the protein is formed at the end of it. So for the formation of peptide bond, rRNA is used, which is example of RNA enzyme, ribozyme. So it is known as ribozyme. Translation is a process that evolved around RNA, indicating that life began around RNA. Since transcription and translation are energetically very expensive process, these have to be tightly regulated. So this is the translation process. So this is the tRNA. It has the anticodons. It has like opposite to it, and when they get joined, it forms a protein. Regulation of transcription is the primary step for regulation of gene expression. In bacteria, more than one gene is arranged together and regulated in units called as operons. So lac operon is a prototype operon in bacteria. So lac operon is a basic operon in the bacteria, which codes for gene responsible for metabolism of lactose. It codes for the gene responsible for the metabolism of lactose. The operons is regulated by the amount of lactose in the medium where the bacteria are grown. Therefore, this regulation can also be viewed as regulation of enzyme synthesis by its substrate. So this is the lac operon. The lac operon has promoter gene, operator gene, and it has Z, Y, and A gene. So Z codes for beta galactosidase and Y codes for permease and A codes for transacetylase. In presence of repressor gene, it binds to the operator region and there will be no translation process will happen. In presence of inducer, it facilitates the process, it transcript to mRNA and translate to form the enzymes. Human Genome Project was a mega project that aimed to sequence every base in human genome. This project has yielded much new information. Many new areas and venues have opened up as consequence of the project. So human genome project, it has like certain points, like there are 3164.7 billion base pairs in a human genome, and chromosome 1 has the most gene and chromosome Y has lower gene. Less than 2% codes for proteins. 50% of gene function is not known yet. DNA fingerprinting is technique to find out variation in individual of population at DNA level. It works as a principle of polymorphism in DNA sequence. So DNA fingerprint is a technique to find out variation in different individuals. It has immense application in forensic science, genetic biodiversity, and evolutionary biology. So it has immense helpful things in the forensic science, genetic biodiversity, and evolutionary biology. The origin of life on Earth can be understood only against the background of origin of the universe, especially Earth. Most scientists believe chemical evolution, formation of biomolecules preceded the appearance of first cellular forms of life. So there are various theories proposed for this evolution: the Big Bang theory, theory of spontaneous generation, and panspermia and all. The subsequent events as to what happened to the first form of life is conjectural story based on Darwinian ideas of organic evolution by natural selection. Diversity of life forms on Earth has been changing over millions of years. It is generally believed that variation in population result in variable fitness. So variation in population, it is the result in variable fitness. So natural selection is the idea of Darwinian ideas is one of this natural selection. Other phenomena like habitat fragmentation or genetic drift may underlay this variation, leading to appearance of new species and its evolution. So there is divergent evolution and convergent evolution. The divergent evolution, it has homologous structures, means they have similar anatomy but has different functions. In convergent evolution, they have analogous structures. They have not anatomically similar structures, but they have similar functions. Homology is accounted by the idea of branching descent. Study of comparative anatomy, fossils, and comparative biochemistry provides evidence for evolution. Among the stories of evolution of individual species, the story of evolution of modern man is most interesting and appears to parallel evolution of human brain and language. So you should know like this adaptive radiation things, like what is the opposite for this placental mammals to Australian marsupials. So if it is mole, it Australian marsupials, it will be marsupial mole. You should know like what other animals replicate the other animals. So human health and disease. Health is not just the absence of disease. It is a state of complete physical, mental, social, and physiological well-being. So health doesn't mean there is no disease. It means physical, mental, social, and physiological well-being of a human. Disease like typhoid, cholera, pneumonia, fungal infection of skin, malaria, and many other are major causes of distress to human beings. So typhoid is caused by Salmonella typhi, and pneumonia is caused by Streptococcus pneumonia or Haemophilus influenzae, and many of the fungal infections and malaria. Malaria is caused by Plasmodium vivax falciparum and malariae, and fatal one is caused by Plasmodium falciparum. If not treated, it may be fatal. Beside personal cleanliness and hygiene, public health measures like proper disposal of waste, decontamination of drinking water, control of vectors like mosquitoes or immunization are very helpful in preventing this disease. So, beside our personal cleanliness and hygiene, like proper disposal of waste, decontamination of drinking water, control of vectors like mosquitoes and all, immunization, like they are very helpful in preventing the disease. Our immune system plays a major role in preventing this disease. When we are exposed to disease causing agents, so immune system plays an important role when we are exposed to disease causing agents. The innate defense of our body, like skin, mucous membrane, antimicrobial substance present in our tears, saliva, and phagocytic cell, help to block the entry of pathogen into our body. So innate immunity is a type of immunity which present at the time of birth. It is not specialized, like saliva, tears, and phagocytic cells are all like general examples of innate immunity. If the pathogen succeed in gaining entry to our body, specific antibodies, humoral immune response, and cell mediated serve to kill these pathogens. So there is humoral immunity and cell-mediated immunity. The humoral immunity includes the antibodies, and the cell-mediated includes T cells and B cells. Immune system has memory. So immune system has a memory. When like first we are encountered by a pathogen, the immunity rate is low. When the same pathogen encounters for the second time with the memory of the T cell, the immunity is fast. On subsequent exposure to same pathogen, the immune response is rapid and more intense. This forms the basis of protection afforded by vaccination and immunization. Among other diseases, AIDS and cancer kill a large number of individuals worldwide. So, AIDS and cancer are the dangerous diseases. AIDS caused by human immunodeficiency virus. So, AIDS is caused by HIV virus and it is fatal. It can be prevented if certain precautions are taken. So, HIV is also sexually transmitted diseases. Many cancers are curable if detected early and appropriate therapeutic measures are taken. So, in cancers, there are two types of tumors, benign tumor and malignant tumor. Benign tumor are localized to a place, but the malignant tumor can pass to different areas. They can lodge there and cause several tumors there. This is known as metastasis. Of late, drug and alcohol abuse among youth and adolescents is becoming another cause of cancer. So among the teenagers, drug abuse and alcohol abuse are becoming very common nowadays. Because of addictive nature of alcohol and drugs, they present benefit like relief from stress. A person may try taking these in the face of peer pressure, examination rate, competition rate, stresses. So why these are is done by teenagers means because of the peer pressure, stress, examination are the major causes. In doing so, he or she may get addicted to them. Education about their harmful effects. Counseling and seeking immediate professional and medical help would totally relieve the individual from the evil. So, education about and counseling to them, like medical help to them, and the friends helping them to overcome may help the individual to come out from the evil. So, this is the structure of antibody. So, this is the antigen binding site. It has light chain and a heavy chain. It has disulfide bonds. So this is the life cycle of malaria. So malaria has two hosts, human host and a mosquito host. In human host, it reproduces asexually. In mosquito, it produces sexually. So the mosquito take up sporozoites. Sporozoites present in the salivary gland of the mosquito. When it bites, the sporozoites get infected inside the humans. So this is the replication of AIDS. Microbes. Microbes are very important component of life on Earth. Not all microbes are pathogenic. Many microbes are very useful to human beings. So not all microbes, எல்லா microbes வந்து harm தான் cause பண்ணும் அப்படிலாம் கிடையாது. So there are certain microbes which are very useful to the human beings. We use microbes and microbially derived products almost every day. Bacteria called lactic acid bacteria grow in milk to convert it into curd. So lactobacillus helps in curd formation. The dough, which is used to make bread, is fermented by yeast called Saccharomyces cerevisiae. So, yeast in dough, idli and dosa batters, and alcoholic drinks. Certain dishes such as idli and dosa are made from dough fermented by microbes. Bacteria and fungi are used in impart particular texture, taste and flavor to cheese. So different cheese has different types of flavor. These are all due to the presence of different types of bacteria. So the Propionibacterium shermani, it produces a flavor to the Swiss cheese. It produces flavor, and the presence of large hole in Swiss cheese is due to Propionibacterium shermani. Microbes are used to produce industrial products like lactic acid. So lactic acid is produced by lactobacillus. Acetic acid is produced by aspergillus. And alcohol is by Saccharomyces cerevisiae, which are used in variety of processes in the industry. Antibiotics like penicillins are produced by useful microbes. They are used to kill the disease-causing harmful microbes. So penicillins are fungi. It used to kill the harmful microbes. Antibiotics have played major role in controlling infections like diphtheria, whooping cough, and pneumonia. For more than 100 years, microbes are being used to treat sewage waste treat by the process of activated sludge formation. And this helps in recycling of water in nature. So many microbes are used in sewage water treatment. There are primary step and secondary step. Methanogens produce methane while degrading plant waste. So methanogens produce methane. So methanogens are a group of bacteria. They produce biogas while degrading a plant waste. So this biogas produced is used as a energy in rural areas. Microbes can also use to kill harmful pests. Process called biocontrol. The biocontrol measures help us to avoid heavy use of toxic pesticides for controlling pests. So this kind of measures help us to avoid toxic pesticides. So there will be no accumulation of pesticides in the plant. There will be no any harmful in the plant growth things and all. There is need to this to push for use of biofertilizers in place of chemical fertilizers. It is clear from the diverse uses human beings have put microbes to that to play important role in the welfare human society. So microbes play a very important role in the human society. So biotechnology deals with large-scale production and marketing of products and processes using living organisms, cells or enzymes. ஸோ வை பயோடெக்னாலஜி இப்போ வந்து ஒரு மெடிசன் ப்ரொடியூஸ் பண்ணோம்னா நம்மளால லைக் வி கான் ப்ரொடியூஸ் வெறும் லைக் ஒரு மெடிசன் அந்த மாதிரிலாம் சொல்லிட்டு ஸோ வி நீட் லார்ஜ் ஸ்கேல் ப்ரொடக்ஷன் நிறையா மெடிசன் நிறையா என்சைம் நிறையா ப்ரோட்டீன் ஸோ ஃபார் தட் திங் வி brought a term called biotechnology. Modern biotechnology using genetically modified organism was made possible only when man learned to alter the chemistry of DNA and constructed recombinant DNA. So major step involved in biotechnology is production of recombinant DNA. This key process is called recombinant DNA technology or genetic engineering. The process involves use of restriction endonuclease, DNA ligase, plasmid, or any other viral vectors to isolate and ferry the foreign DNA in the host organism. So this involves series of reactions like first ஒரு plasmid வேணும் and it has a restriction endonuclease. Restriction endonuclease தான் வந்து help to like cut பண்ண யூஸ் ஆகும். Ligase is used to attach the specific gene to the plasmid. அப்புறம் it is incorporated in the host organism. Expression of foreign gene, purification of gene product. Large scale production involves use of bioreactor. So இன்னும் like if we need to produce more, we use like structures called bioreactors. So this is how recombinant DNA is formed by restriction endonuclease E. cori. So the E. cori cut at a specific site where the bases are GAATTC. So both the vector DNA and foreign DNA are cut with the same restriction endonuclease, so they can have the same bases. Then the sticky end get attached by DNA ligase to form a recombinant DNA. So this recombinant DNA is like made to keep with the host organism. So this is the foreign DNA, vector DNA or plasmid. So the rate restriction enzyme cut at a specific point used to join by DNA ligase, then it get incorporated into the cell. So for producing more, like producing more gene, more copies of gene, there is a process called PCR. PCR has 3 steps: denaturation, annealing, and extension. So first denaturation, the both the strands to be separated by high heat, like 72 degree is used there. Then annealing. At the 3' end, RNA is added for annealing. Then extension of the RNA strand. So this gives the amplified genes. So biotechnology has given humans several useful products by using microbes, plant, animals, and their metabolic machinery. Techniques of tissue culture and somatic hybridization offer vast potential for manipulation of plants in vitro to produce new varieties. Recombinant DNA technology has made it possible to engineer microbes, plants, and animals such that they have novel capabilities. Genetically modified organisms have been created by using methods other than natural methods to transfer one or more genes from one organism to another. So genetically modified organism, they have been created genetically modified organism by using methods other than natural methods, and they transfer one or more genes from one organisms to the another. So this technique is called recombinant DNA technology. So genetically modified plants have been useful in increasing crop yields, reduce post-harvest losses, and make crops more tolerant to stresses. So these genetically modified plants, they increase crop yields, reduce post-harvest losses, and make crops more tolerant to the stress. There are several genetically modified crop plants with improved nutritional value of food and reduce the reliance on chemical pesticides. Recombinant DNA technological process have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutics. So DNA, this recombinant DNA technology, it has like major place in the healthcare by producing mass therapeutics. Since the recombinant therapeutics are identical to human proteins, they do not induce unwanted immunological response and are free from risk of infection, as was observed in case of similar products isolated from non-human sources. As these therapeutics are very similar to human proteins, they do not cause any harm to the humans. Human insulin is made in bacteria, it's structurally absolutely identical to that of natural molecule. So this is how insulin is made. So this is how pro-insulin looks like. So the A peptide and C peptide is separated from the free C peptide. Transgenic animals are also used to understand how genes contribute to development of disease by serving as models for human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Alpha-1 antitrypsin for emphysema has been developed, and then high protein yielding cow milk are also been developed and all. Gene therapy is the insertion of gene into individual cells and tissues to so to treat disease, especially hereditary diseases. So gene therapy, it is called insertion of gene into an individual cell to treat diseases. So first it was done with adenosine deaminase deficiency. This gene is inserted in the lymphocytes and it is allowed to multiply. It does so by replacing a defective mutant allele with a functional one and gene targeting, which involves gene amplification. So in ADA deficiency, there is a mutation of one gene that produces adenosine deaminase. So gene therapy is used in this case. Viruses that attack their host and introduce their genetic material into the host cell as part of their replication cycle are used as vectors to transfer healthy genes or more recently portions of genes. So the viruses which attack the host, which introduce their genetic material into host cell, are now used as vectors to transfer healthy genes to the host. The current interest in the manipulation of microbes, plants, and animals has raised serious ethical questions. So that's all. So more than summary, full lesson படிச்சுட்டு like if you like, get to see summary, you will get like இதெல்லாம் படிச்சிருக்கோம் அந்த லெசனில் like that. Don't completely depend on summary and all. Read the lesson like line by line in from NCRT and look at the summaries and all. Get this test series, like practice more and more. So ஒரு லெசன் படிச்சுட்டு if you get use this test series, it will be like very useful. Like it has like every chapter wise questions, grant test and all. அதுவும் வந்து like it's available for வெறும் 500. So it will be very useful. Kindly make use of it. ஸோ அவ்வளோதான் பாட்னி அண்ட் ஜுவாலஜி சம்மரி பாக்ஸஸ் அண்ட்